Hey guys, and welcome back to Now I Know. Today we are continuing our discussion on hypersensitivity reaction. In the last video we saw what is hypersensitivity reaction and type 1 hypersensitivity. Now briefly, what is hypersensitivity? It is increased or inappropriate immune response. Either it is way too high or immune response where actually it is not needed. And today we are going to see type 2 hypersensitivity, that is called antibody mediated cytotoxic hypersensitivity. Long name but easy to understand. It is the type of hypersensitivity which is antibody mediated. Okay, it is called cytotoxic because see, generally what happens when you have an antigen in our body, when antibody binds to it to clear it up, it forms antigen antibody complex, right? And this complex can actually call so many other things for help, like it can activate the complement system. It can also call other cells like NK cells, neutrophils, macrophages to clear up the antigen. Now, if complement is called, we have already talked about the whole complement system. It results in membrane attack complex, which eventually, which will lyse the cell. So cell lysis occurs. Okay, then you have neutrophils or macrophages or natural killer cells. They are also cytotoxic. Let's say, for example, neutrophils can degranulate and produce the toxic substances that can also cause lysis of cell. So there is a cytotoxicity. The death is by cytotoxicity, and it is because antibody is bound to the antigen. That is why it is called antibody mediated cytotoxic hypersensitivity. So that's why hypersensitivity 2 is called antibody mediated cytotoxicity, because as I said, the problem is when your antibody is causing destruction of healthy cell. Now this normally that's what happens. You have antigen antibody complex made, and that causes lysis of cell because it can activate complement system, it can call other cells and lyse the antigen. That's the normal way how our immune system is going to fight against the antigen. But when the same thing happens against your own cell, against the healthy cells which are part of your body, that is where the problem occurs. That is what is type 2 hypersensitivity, where the antibodies, and mainly when we talk about type 2, it is IgG. Mostly you will see IgG mediated type 2, or sometimes it is also IgM. So these antibodies are against self antigen, which is part of your own body, which is actually not harmful. So it is against the intrinsic antigen, that is part of your body, self antigen, that is present on your own cell. Let's say, for example, on RBC, the molecules on RBC can get recognized as an antigen. Antibodies would come and bind and will cause lysis of the cell. Now this is your own cell, you don't have to do that, you don't have to kill your own cell. But in type 2 hypersensitivity, this is what happens. So that is what is called intrinsic antigen, self antigen on your own cell. Now, other than this intrinsic antigen, there is also extrinsic antigen. That means an antigen that has come from outside of a cell and that will attach to your own cell. Let's say, for example, if you know about some drugs, some haptens. Haptens can, bind to your own cell and then it will act as an antigen. In that case, let's say, for example, penicillin is such a drug that would bind to your own cell and then it will be recognized as an antigen. Because it's an hapten, it's a very small molecule, alone it is not going to get recognized. But when it will bind to your own cell, let's say, for example, it has bound to your own cell, now this becomes antigen. Okay, so your body is going to mount an immune response against your cell only because the antigen or the hapten in this case is bound to your own cell. So what you're destroying is your healthy own cell. So whether it is intrinsic or extrinsic antigen, whether it is your own or it has come from outside and attached to your cell, the final thing that your immune system is doing is killing or destroying your healthy cells. So that is what happens in case of type 2 hypersensitivity. Alright, antibody mediated lysis of your own healthy cell, and we will see what and how it happens. Now that's what, as I said, in normal infection, antibody is complex to antigen and you're going to destroy that outside antigen. But in case of hypersensitivity 2, your antibody is going to destroy the host tissue, because in case of extrinsic antigen also it is bound to your own cell. So finally what you are destroying is your own tissue. Okay, so that's the difference. In case of normal infection, antibody is bound to antigen, so you're killing an antigen. But in case of hypersensitivity to, antibody is actually bound to your own cell and it is going to kill the healthy tissue or your own cell. Now let's see how and what happens. So the first type of type 2 hypersensitivity that could occur is because of the complement mediated reaction. Because what happens is in normal condition is when antibody is bound to an antigen, we have talked about all the complement pathways and we know how antigen antibody complex can trigger complement system. There will be cascade of complement reaction where C1 molecule first will come and bind to your antigen antibody complex, and that how it will result in membrane attack complex. We have talked about this in detail. So complements will get activated if there is antigen antibody complex, right? In this case, if there is let's say for example your own cell is bound by an antigen, extrinsic antigen. Let's say for example penicillin, drug penicillin can bind to your own cell. Now this for the immune system becomes an antigen, even though it is not. But in case of type 2 hypersensitivity, this is going to be recognized as an antigen. So it's a target cell for the immune system. So against this antigen, immune system is going to form antibodies. Alright, so it will coat the target cell. Antibodies will coat the target cell. Now once the antigen antibody complex is made, it is going to activate the complement system. One way is that the MAC formation, where the C1 to C9 all cascade reaction will occur and there will be formation of pore on the surface of antigen. If you have not seen the video and if you don't know about this mechanism, just have a look on the complement pathway. But what happens essentially after the complement pathway or after the complement system is activated is the biggest outcome of complement system is formation of membrane attack complex. That is nothing but formation of pore on the antigen. Now because there is pore, the contents of the cell will go out, something from outside will enter the cell, so there will be loss of osmotic balance. As a result, there will be cell lysis. So this cell is now killed. Now what was the fault of this cell? This was our own cell. Only some drug from outside had come and attached to this cell. This was not actually an antigen, but now this acted as an antigen because hapten was bound to my own cell. Now this is one way, MAC formation. Other than that, if you remember, we had talked about when the complements get activated, there will be splitting of complement in the A fragment and B fragment. So out of this, when there is splitting of the fragment, the C3b molecule, complement 3b molecule, act as an opsonine. That means it can go and bind to the Fc portion of this antibodies that is coated. Look at here, all the C3b molecules have bound to this Fc portion. Why is it doing so? Because it is opsonizing the cell. That means it is marking the cell and telling the macrophages that this is the cell you need to eat up. So opsonization is a process of making the cell ready for phagocytosis by the phagocytes. So once this C3b molecule, that is nothing but broken down product from complement itself, will go and bind to Fc portion of all the antibodies that has coated this antigen, that would result in opsonization followed by phagocytosis. Macrophage doesn't understand whether it is self-cell or not. All it will see is, because it has C3b receptor, it will see that there is C3b bound on the cell, it will go and carry out the phagocytosis. So again you are killing the own cell. Now other than that, once the complements are getting activated, as I said, there will be fragmentation in A and B fragments. So the C3A, C4A, C5A, these fragments actually, once they are formed, they act as attractant for neutrophils. Okay, so once these fragments are formed, they will act as attractant and it will call neutrophils at this site. Now neutrophils have Fc receptor on their surface. Like as I said, macrophages have C3b receptor. Neutrophils would have Fc receptor on the surface. So what happens is, this can recognize the Fc portion of bound antibody on the antigen, and this binding will result in degranulation of the neutrophils. And once they will degranulate, it will release the substances like peroxidase and myeloperoxidase that can generate the oxygen radicals which are toxic to the cell, causing the tissue damage. Again, it is going to kill the cell. Okay, so this is what happens once the complement is activated. One way is membrane attack complex, pore formation, lysis of a cell. Then C3b molecule, C3b fragment, which is a very good opsonine that will opsonize the target cell, resulting into phagocytosis by macrophages. Also broken down product of complement system like C3A fragment, C4A, C5A can act as attractant that would attract neutrophils. Since they have Fc receptor, it will bind to the Fc portion of bound antibody, resulting into degranulation, releasing toxic substance, eventually killing the cell. So that's how complement 3 ways can damage your own tissue. So that's how in case of hypersensitivity to complement mediated reactions occur. Now other than complement activation, what else could happen? So the next type is called antibody dependent cell mediated cytotoxicity. You might have heard about ADCC, antibody dependent cell mediated cytotoxicity. That means there is no complements involved, but the cytotoxicity is dependent on antibody. Once the antibody is bound to antigen, it will result in the killing or lysis of the antigen. Let's say for example, the antibodies, mainly it will be IgG mediated. Okay, so the antibodies are bound to your self cell, some molecules on the RBC. Now this antigen antibody complex, this binding, now the cells, natural killer cells, macrophages, neutrophils, eosinophils, they have the receptor for the Fc portion. So it's very simple to understand. This Fc portion would bind to this antibody that is bound to antigen. In this case it is self cell. This binding will result in a degranulation, release of toxic granules, and these granules contains two important things. One is perforins that can cause a pore formation on the cell, and through this pore granzymes would enter in the cell, causing apoptosis of the own cell. Okay, this is a cytotoxic effect mediated or because of the antibody it occurs. So it is called antibody dependent cell mediated cytotoxicity. All these cells like natural killer cells, macrophages, neutrophils, eosinophil can carry out this process because they have the granules containing the perforins and granzymes. Now this was about the antibody mediated cytotoxicity. The next one is antibody mediated, but it is not cytotoxicity, it is a cellular dysfunction. Okay, it is causing cellular dysfunction which is non-cytotoxic, but it is antibody mediated, where what happens is antibodies will bind to some of the cell surface receptor, and these receptors are very important to carry out certain functions. Let's say for example, it can either pass the nerve impulses, or it is important for production of some hormones. So these receptors, once these antibodies bind to this receptor, it will either block the function of that receptor, or it will cause overproduction of some hormone. Let's say for example, and I'm sure you must have heard about this or you must have read about it, In case of myasthenia gravis, what happens is the acetylcholine receptors that are important for binding of acetylcholine and muscle activation. There you have antibodies, auto-antibody production that would go and bind to the acetylcholine receptor on the muscle cell. As a result, it will block the acetylcholine binding and it will result in impaired neuromuscular transmission, resulting in the weakness of the muscle. That's how it is blocking. Auto-antibodies are blocking the some function. It is stopping or it is inhibiting some function. On the other hand, it also works in other way. In Graves' disease, what happens is auto-antibodies are produced against the thyroid stimulating hormone receptors on the thyroid epithelial cells. Okay, in normal condition there will be regulated production of thyroid hormones, but in case of Graves' disease you have auto-antibodies against the thyroid stimulating hormone receptors on the cells, on the thyroid epithelial cells, and once this binding happens, antibody will bind to this receptor. It gives signal for the production of thyroid. Overproduction of thyroid hormone would occur, resulting into hyperthyroidism. So on one hand you saw it blocks the function, on the other hand it causes overproduction of hormones. So that's how auto-antibodies can result in the dysfunction, cellular dysfunction. So antibody can mediate cellular dysfunction, which is non-toxic. On the other hand, it can also cause cell-mediated cytotoxicity. So antibodies can cause damage in two different ways: cytotoxic and non-cytotoxic, and complement can cause problem in three different ways, as we saw. Now this was all about the mechanism how type 2 hypersensitivity can cause problem, but let's see some examples here. When it comes to cellular dysfunction, these are the two examples. Myasthenia gravis and Graves' disease are two examples of antibody mediated cellular dysfunction, but let's see some other examples of type 2 hypersensitivity. Now let's say, for example, in case of transfusion reactions, you will observe type 2 hypersensitivity if there is ABO incompatible blood. Let's say, for example, a person is blood group O. That means the person's RBC doesn't have antigen A or B on its surface. So if you give such a person blood group A, now since the person's body doesn't have this blood group A, it is going to recognize this as an antigen. Even though if you think about it, it's just a blood group, but this would be recognized as an antigen by this blood group O individual. So this person will mount immune response against the newly entered blood group A cells, and it will cause the complement mediated cytotoxic reaction. Okay, so transfusion reactions, if you have incompatible blood group, it will result in complement mediated cytotoxic reaction, that is type 2 hypersensitivity. Another example is a drug induced reaction. As I said, some drugs like penicillin, they actually act as heptane. Okay, only when they combine with some of our own cell will be recognized as an antigen. So some drugs or some heptanes, when they combine with our own cell, they would be recognized as an antigen by our immune system, and it will cause antibody formation. Once the antigen and antibody complex is formed, what will happen? It will result in cytotoxic damage, as we just saw. That once the penicillin is bound to cell, this is now an antigen. This target cell would trigger the antibody reaction. Antibody will be bound to it. There will be complement mediated lysis of this target cell. So drug induced reaction where the heptane will bind to our own cell, resulting in that would be recognized as an antigen, would result in cytotoxic damage. So that is also an example of type 2 hypersensitivity. Another example of type 2 hypersensitivity is RH incompatibility, and this is also one of the topics that we have talked about it. RH incompatibility is also known as erythroblastosis fetalis or hemolytic disease of the newborn. And it is a condition when Rh negative mother has Rh positive fetus, and at the time of first delivery, when there is mixing of the fetal blood with when it enters the mother's circulation, since the mother is Rh negative and the fetal blood would have Rh positive cells, it will be recognized as an antigen by mother's immune system, and it will try to, and not try, it will eliminate, or it will clear up this Rh positive antigen, creating the memory B cells. So in the subsequent pregnancy, what happens if the fetus is Rh positive? Now the antibodies that are made against this Rh antigen is IgG, and as I said in the beginning, type 2 hypersensitivities are IgG antibody mediated. Now IgG can cross placenta in the subsequent pregnancy, and it will cause problem for the fetus because there are Rh positive cells, so it will react against it, causing the lysis of RBC. That is why it is called the hemolytic disease of the newborn. So in this case, there is a preparation of anti-Rh antibody given after the first delivery and all that. You can have a look if you want in the Rh incompatibility video, but this is what essentially happens. And the last is reaction involving some of the cell membrane determinants. Let's say, for example, on our own RBC there are certain molecules. Now there can be various reasons, maybe due to some infection or due to any unknown reasons. You might have some autoantibodies that are made against your own blood cell. Okay, let's say for example RBC, autoantibodies against your own RBC due to any defect. So if the antibody would be bound against the RBC, it can either cause the complement mediated lysis of the cell, so RBC hemolysis could be there, or it can cause phagocytosis resulting in progressive anemia. This is something what I showed you before. If there was a hapten over here, if the penicillin molecule was bound over here, same thing would happen, right? But in this case, it is actually autoantibody, the antibodies against your own cell, that is causing the same effect. So either it will be complement mediated destruction or your phagocytic cell, because they have the Fc portion receptor. So either it will be direct complement mediated destruction, or because the phagocytic cells would have the Fc receptor, it will cause lysis of the blood cell resulting in progressive anemia. So these are some of the examples of type 2 hypersensitivity. The type 2 hypersensitivity becomes a problem because the antibodies that are produced are either against our own cell, and that antigen antibody complex is being recognized by our cell, and the antibody here, as I said, is IgG mediated. That's why you have Rh compatibility also falling under this category. So because your antibodies are, autoantibodies are against your own cell, is why type 2 is a problem. Now there can be also extrinsic antigen. As I said, in case of blood transfusion, it is from outside. The blood group which is not matching is from outside, or some haptens which has entered our body will complex with our own cell, could be triggering type 2 hypersensitivity. Since it is antibody mediated, it is called antibody mediated cytotoxic hypersensitivity. So that's all about type 2 hypersensitivity reaction. We have seen all the mechanisms and some of the examples. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I will see you next time. Until then, keep learning.